Hello viewer, today I will discuss about the physical mutagen. I will talk about the introduction, the definition, then I will talk about characteristics, sources, type, discussion of each type, biological effect, clinical effect, penetration power, measurement, application as well as effect on environment by physical mutagen. First one, general introduction. So, these high energy containing physical mutagen can penetrate through the cell and causes genetic effect. So, the effectiveness of such high energy radiation or physical mutagen is directly proportional to the penetration power. Next, so physical mutagen are the mutation causing agent including the physical causes like heat, like radiation as well as microwave, and usually the radiation is related with this sort of damage or effect. So, it is called radiation mutagenesis as well as sometimes it is referred to as radiation genetics. Next, characteristics. So, physical mutagen can directly damage to DNA. It can causes the chromosomal abnormalities like translocation, deletion, etc. or it can causes the damage to the bases or base pairing. Ultimately, it is manifested as phenotypic observation. Next one, sources of radiation. It may be natural sources or artificial sources. As natural sources, it was found that background radiation from that of the radioactivity from soil as well as atmosphere as well as cosmic ray and sun ray which contains ultraviolet radiation. And as artificial source, it includes medical testing and nuclear testing. The diagnostic X-ray is often referred to as the artificial source of mutation, and radioactive decay from 14C carbon which is getting converted to nitrogen. Types. Mainly three types are there: heat, microwave and radiation. The radiation may be mainly particulate or electromagnetic. The particulate radiation includes alpha and beta ray, and electromagnetic radiation includes ionizing and non-ionizing radiation. Non-ionizing radiation is ultraviolet radiation and ionizing radiation includes X-ray, gamma ray and fast and thermal neutrons. First one, the effect of heat. Heat cleaves beta-N glycosidic linkage in case of bases, giving rise to the AP site, apurinic or apyrimidinic site. Heat can increase the mobility, so it gives rise to the DNA breaks, translocation, inversion, such kind of chromosomal damage. And heat also helped to convert cytosine to uracil, causing the Cg2Ta transition. Sometimes it can cause transversion also. Next one, effect of microwave. 1 hour exposure to microwave is equivalent with 1 hour exposure to X radiation. Now, radiation, it includes the particulate radiation, include alpha and beta ray, electromagnetic radiation including X-ray, gamma ray as well as fast and thermal neutrons. So, characteristic includes the positive and negative particles coming out from radioactive unstable nuclei. Types includes alpha ray as well as beta ray. First, in case of alpha ray, so I will talk about characteristics and mode of action. As characteristics, we can say the alpha ray is the positive particles, okay. They are coming from the unstable radioactive nuclei of radium, actinium, thorium, etc. And as mode of action, we can say it shows the bystander effect. That means unirradiated cell show the effect of irradiation from neighboring irradiated cell, and it actually repels the negative part of the DNA, causing DNA damage. Next one, beta ray. As characteristics, we can say it is coming from 3H, 32P or 35S unstable radioactive nuclei. Beta is the negative particles, and this beta causes excitation ionization to give rise to high energy emission from the atom. And as mode of action, we can say as it is negative particle, it repels the positive part of the DNA, and it causes transition, translocation, inversion, etc. Characteristics and types. As characteristics, we can say electromagnetic radiation is the proper energy. It propagates via wave. Wave may be longer or shorter. Shorter wavelength contains higher energy. And we can find here, in case of electromagnetic radiation, it is electronic wave as well as magnetic wave. Now, types, here we can say mainly they are ionizing and non-ionizing. In case of ionizing radiation, first general characteristics, then types. As general characteristics, we can say these ionizing radiation as X-ray and gamma ray, they can propagate the reactive ions production by interacting with biological molecule. Each photon contains several electron volt of the energy. So, ultimately they are interacting with the biological molecule coming from unstable radioactive nuclei. In case of X-ray, I will talk about the characteristics, mode of action, genetic effect and nature of effect in different systems. The first one, X-ray characteristics. According to the inventor scientist named Röntgen, it is termed as Röntgen radiation. What happens? Suppose electron violently hit the DNA, then it goes to the lower energy level. So, it dissipates the energy as X-ray. It contains 120 kilo electron volt frequency and amount of energy. The frequency of the radiation is 30 exahertz to 30 petahertz, and we can find it contains enormous amount of energy in case of X-ray, which can penetrate through the biological material, and it has 10 to 0.01 nanometer of wavelength. Next one, mode of action. It includes the radiation intensity as well as chemical effect. First, the radiation intensity. Weak X-ray over long period of time is called as chronic effect or chronic irradiation, which gives the same effect to the strong X-ray for short period of time, called as acute irradiation. So, chronic and acute irradiation both shows the same effect. As we know that by single hit kinetics, for point mutation or recessive or sex-linked lethal, the single dose is needed. And as we know that dose and survival relationship is linear. But as the doses are increased, suppose for 2 hit kinetics, when we need it for the chromosomal abnormalities or breakage, so the square of the dose is needed. And for multiple hit, we show the mutation efficiency is lowered by 3/2 power. And this sort of dosage is independent of the wavelength. And we can find in case of these radiation intensity, the mutation frequency is also independent to the radiation intensity. Next, chemical effect. The chemical effect can be manifested either by direct effect or by indirect effect. Direct effect means the radiation is directly damaging the biological molecule, as is manifested in ion pair formation, or propagate via free radical formation, or indirect effect can also be manifested by the propagation through the water molecule. So, that is the direct effect. Now, in case of indirect effect, we can find the presence of the free radical formation. So, what is going on? H dot and OH dot is getting produced, which along with the presence of oxygen gives rise to HO2 dot hydroperoxyl radical. Now, this hydroperoxyl radical ultimately can be converted to H2O2, hydrogen peroxide. These hydrogen peroxide along with other organic peroxide can actually help to damage the DNA as well as other biological molecule. Genetic effect of X-ray. It is manifested as breakage of hydrogen bond, losses to the bases, damage to the bases, intra and interstrand cross-link to the DNA, as well as single strand and double strand breakage of the DNA. The effect of X irradiation can be increased with the help of presence of oxygen. Next one, nature of damage of X-ray in different systems. I will talk about the effect in case of the Drosophila as well as in case of mice. Number 1, in case of Drosophila, it was found that the mutation rate of point mutation is directly proportional to the dosage applied, and it is called single hit kinetics, showing the exponential kinetics, and survival fraction is to dose, shows the linear kinetics. No split dose, no safe dose is present, and split doses are cumulative. Here we can find the system operates some sort of selection and repair, but selection and repair, it is actually related to the time interval, and we can find the presence of other effect like other dose related kinetic, and dose is actually independent to the radiation intensity, as well as these radiation intensity or dose is not dependent on the wavelength, but dosage remaining constant. And we can see in case of Drosophila, apart from this sort of relationship, no time is actually dependent on these kinetics, and the phenotypic observation is related to the CLB technique of the observing phenotype, that is related to the sex-linked recessive lethal. C stands for crossover suppressor, L stands for recessive lethal, and B stands for bar-eyed phenotype. So, in case of irradiated male, the CLB technique is found to mate with the female having proper proportion in the next generation. So, in this way the X irradiation effect can be monitored. Next one, in case of mice, it was found that the chronic doses shows a fewer amount of the mutation rate than that of the acute doses. In case of intermittent dose also, fewer mutation rate is found than that of the continuous dose, though in both the cases total doses are same. The repair system is found in case of oocytes, not in sperm, and we can find the repair is dependent upon the cellular condition. The mouse 7 locus test give the proper database, and we can find here also no time is dependent on the kinetics, and rarely the single hit kinetics is found because some robust repair system is actually found over here. Next one, gamma ray. Gamma rays are coming from subatomic particle from the cyclotron, and it is the characteristic. As mode of action we can find in case of MUBIN, the gamma ray causes the reduction in the germination and causes lethal dose LD50. Fast and thermal neutrons. Fast neutrons are coming from the cyclotrons. If their velocity is retarded or slower, that causes the formation of thermal neutron. So, that is the characteristic. As mode of action we can say it can damage to the biological material because they are highly densely ionizing radiation. Number 1, characteristics, it has a wavelength from 200 to 400 nanometer, and the amount of energy is 3 electron volt to 124 electron volt. Ultraviolet radiation is absorbed by purine and pyrimidine at 254 nanometer, and it is less penetrating than X-ray. It causes the formation of H plus and OH minus and causes thymine-thymine dimer formation. Mode of action, direct effect or indirect effect. Direct effect causes the formation of thymine-thymine dimer as well as DNA strand breaks. Number 1 type, it includes the CPD, cyclobutane pyrimidine dimer, that is actually come from the C double bond C, and it actually give rise to the covalent bond formation called as pre-mutagenic lesion, but that can be repaired by excision repair and photolyase repair. 6,4 photoadduct, another type of photoadduct formed from direct damage to the UV radiation of the DNA, and it is formed from 6,4 pyrimidine pyrimidone complex. It is hydrolyzed by spore hydrolyase, but it is not frequent, that it is found 1/3 than the former, but it is causing the much more mutation. Next, thymine-thymine dimer causes the perturbation of the DNA double helix. So, it stops the replication and transcription, causing the error. Next one, DNA strand breaks. UVB causes strand breaks of the single strand as well as double strand, and this causes the lethality. Next, indirect damage can be propagated by the intermediate absorption compound as well as the formation of free radical. And pre and post treatment. The pre-treatment of the culture medium in case of Staphylococcus aureus with UV radiation give higher mutation frequency in the unirradiated cell, and the post-treatment with the chloramphenicol can decrease the mutation rate. Next, the phenotypic expression in case of Drosophila, E. coli and Aspergillus. In case of Drosophila, it was found that in polar cap cells, the effect was found by Altenberg first. Then the polar cap cells are present in egg and it was extrapolated to other cells. But if UV ray have little penetrating power than that of the X-ray. Next one, in case of E. coli, streptomycin resistant E. coli shows the single hit kinetics with respect to UV dose. And in Aspergillus species, the morphological mutations are found in case of UV. Dose kinetics, the dose response kinetics is dependent on the type of mutation, the condition as well as organism. And here we can find in seldom the UV related single dose kinetics. Action spectrum, the radiosensitivity versus wavelength curve is called action spectrum. Action spectrum of DNA is equivalent to the UV radiation. And in case of bacteriophage, with respect to the wavelength, some single hit kinetics graphs are found where different slopes are observed. Next, the clinical nature of UV damage. Number 1 is the acute effect causing erythema, that is sunburn, transient effect, that is immune response, and the chronic effect as skin cancer. Then penetration power of radiation, the penetration power is inversely proportional to the wavelength, and beta particle has much more penetration power than that of the alpha particle for its small size, and the neutron having no charge is most penetrant. There are so many different types: systematic effect, effect on dividing and non-dividing cell, germline effect and chromosomal effect. Systematic effect in somatic cells, it causes the carcinogenesis, and in case of genetic effect, it shows the abnormalities to the next generation. In case of chromosomal effect, we can find in case of interphase, the breakage happens to give rise to the centric and acentric fragment by breakage near centromere. And in S phase, both the fragments are joined, but in case of anaphase, these are joined to the spindle fiber. So, it inhibits further division, so it gives the giant cell formation and inhibiting further nuclear division. Next one, in dividing cells like RBC, like here, so much more radiation effect is found than rarely dividing cell like muscle, bone, etc. In germ cell, it stops the cell cycle and giving rise to the cell death by arresting the cell division. That is related to single gene disorder, related to X-linked recessive and dominant lethal, chromosomal disorder, multifactorial disorder related to the oncogene activation and suppression of tumor suppressor gene, as well as it was found in case of deterministic effect when large amount of radiation kills large amount of cell, and prodromal effect after nuclear disaster, which was found in case of Chernobyl in 1986, as well as it was found in case of Hiroshima and Nagasaki, where after nuclear bombing the effect was found after several generations where teratogenic babies were born. It was found in plant and in case of animal. In case of plant, it actually giving for crop improvement by mutation breeding. The radiation therapy is done in animal against the cancer as radiation therapy. Next one, the impact of mutagen over the environment. The impact may be the positive impact or negative impact. As negative impact, it may be in plant, animal or in marine life. In case of plant, we can observe the weakening of the seed, retarded germination. In case of animal, we can find the abnormal birth for several generations as well as cancer, and in case of marine life, it gives to the retarded reproduction power in case of animal system, and in case of plant system, it actually gives the different later timing of the plant flowering. So, plant flowering is getting delayed here. Next one, the positive impact in case of some non-ionizing radiation. It helps the plant for its growth and it also helps them for photosynthesis. So, students, I have discussed about several ionizing and non-ionizing radiation, their effect, their doses, clinical symptoms, their effect over the environment, their penetration power as well as their measurement strategies. Thank you.